Hello, I am Professor Sims, and this video is going to be covering laboratory diagnostic techniques for pathogenic microbiology. This is the fourth of 10 lessons included in my pathogenic microbiology course. If you're currently enrolled in this course, please consult the syllabus and the course Moodle site for assignments and other course information. The learning objectives for this unit are going to be comparing characteristics of monoclonal and polyclonal antibodies, describing various types of assays used to find antigen-antibody complexes, comparing direct and indirect agglutination, identifying various uses of hemagglutination and diagnosis of disease, how blood types are determined and used in transfusion. We're going to talk about EIA, FEIA, and ELISA between immunohistochemistry and immunocytochemistry. We'll talk about direct and indirect fluorescent antibody assays and how flow cytometry is used to quantify subsets of cells present in complex mixtures of cells and cell types. The specificity of antibodies makes them a really good tool to use for detecting and quantifying targets, and this has to do with the science of making drugs and finding new ways to diagnose disease. So what you do is you precipitate the soluble antigens, or you can agglutinate cells, or you can opsonize, kill bacteria. The antibodies, they bind with very high specificity to the antigens, but they also, and we've seen this already in the earlier lessons, they also show some cross-reactivity by binding to antigens that have similar chemical properties than their targets. You can inject an antigen into an animal, and what that is called is polyclonal antibodies. That's how you develop polyclonal antibodies. Due to the possibility of false positive or negative results, polyclonal antisera are typically only used for just screening tests. Monoclonal antibodies are much more specific because they only bind to a single epitope and they have very, very high affinity, especially compared to polyclonal antibodies. If an antigen has one protein and multiple epitopes, you can use that to stimulate production of many antibodies, some of which may bind to identical epitopes, and this is what is referred to as cross-reactivity. Polyclonal antibodies can be obtained by injecting lab animals with an antigen, and what happens next is the animal's own immune system will make a whole bunch of antibodies that are specific to that antigen. And then when you harvest those antibodies from the animal, that is what is called antiserum. So that's what they're showing here in figure 23 or 20.3. So you inject this rabbit here with an antigen, and his B cells will come and attach to the epitopes of that antigen. It produces memory B cells and plasma B cells, and those memory B cells will secrete antibodies, and in that way you can get a lot of polyclonal antibodies from a small amount of antigen. So in order to help to kind of differentiate these two terms, polyclonal, monoclonal, so I've got a summary here of each type of antibody production. Your polyclonal can be of many different species. It's less specific. There can be very different affinity from one batch to another. You are more likely to have success in an unknown application. So what that means is happy accidents can happen with polyclonal antibody production. Monoclonal, it usually is cleaner. Your antibodies are going to be really consistent from batch to batch, but you're more likely to get false negative results. So let's look some more at monoclonal antibodies. These are produced by introducing an antigen to an animal and then fusing the polyclonal B cells from the mouse, in this case the mouse, to myeloma cells. And what happens is you have what is called a hybridoma. Hybridoma cells have been cultured. They continue to produce antibodies, and hybridomas that are producing the desired antibody are grown out in large numbers on a selective media. And you can periodically harvest those media to get the antibodies that you want. So there are extra steps involved in monoclonal antibodies, but it allows you to get antibodies that are much more specific. Okay, this is another figure comparing polyclonal to monoclonal antibody productions. As antibodies bind more and more antigens together, it forms a precipitin, which is basically just a big clump of antigens and antibodies. And this is happening in what is known as a lattice formation. And you can use this in order to get those antigens and antibodies to precipitate out of a solution. And this is just showing how as more and more precipitin is formed, you get to this equivalent zone. And then if even more and more precipitin is formed, you have a zone of antigen excess. This is figure 20.6 that says, as antigen is slowly added to solution containing a constant amount of antibody, the amount of precipitin increases, and the antibody to antigen ratio approaches the equivalent zone and then decreases once the portion of antigen exceeds the optimal ratio. So it can be kind of tricky to try to perform an experiment where you're adding antigen, you're adding antigen, and then you hit a sweet spot and you want to stop. You want to stop adding antigen because if you get to this point here, then you won't have enough antibody, right? The antigen kind of takes over and you have a lot of precipitin, you have a lot of antigen, and not enough of the antibody that you're trying to isolate. A precipitin ring test is when you use a standard antigen solution in the bottom of the tube, and then you do a serial dilution of antiserum on the top. You use glycerol to prevent the two solutions from mixing because glycerol is very dense, right? And the precipitation only occurs at the interface of the glycerol. A visible ring of precipitation is seen at the 1/4, 1/8, and 1/16 dilutions, indicating these concentrations are within the equivalent zone. So I used to do a lot of research with viruses, and they're calling this the precipitin ring test. When I did it, I used to call it glycerol precipitation. And essentially, it's you trying to separate out the antibody, and you're utilizing glycerol in order to do that, to keep the solutions separated. And then you can determine the optimal amount of antigen to antibody ratio by doing a serial dilution, right? So if you have a 1-to-1 antibody antigen, 1-to-2, 1-to-4, 1-to-6, 1-to-16, and 1-to-32, and the precipitin will form where the glycerol layer is, and based on how much of precipitin you have, when you start to see it at all, okay, then you know you're getting close to the equivalent zone. And then once you have a nice solid ring of precipitin, you know that is the dilution that you want to continue to work with in your studies. The precipitin ring test has some drawbacks. It does require a lot of the serum, and it is kind of difficult to avoid mixing the solutions and disrupting the ring. You got to be real steady-handed with these tubes. An alternative is doing the test in an agar gel. This is called double immunodiffusion. It minimizes these problems, and it allows for really rapid detection of antibody-antigen ratios. And it's really useful when you're looking at situations where you may have cross-reactivity, because you can check the antiserum against closely related antigens and see which combinations do and which ones don't form these precipitin arcs. So this is what it looks like, this kind of assay. It's an Agglutination test, and what happens is you place an antigen and the antisera in a gel, and the antibodies and antigens will diffuse through the gel, and this causes a precipitin arc to form when you have reached the correct ratio to have what is called a zone of equivalence. In this example, the interserum in well 1 contains the antibodies to the antigen, and then the resulting precipitin arc is stable because the lattice is too large to diffuse through the gel. So this is showing what's happening at the precipitin ring. You have the antigens are in this well, you have the antibodies that are in this well, and this This is where the antigens and the antibodies are coming together and binding to each other, and they're forming a solid precipitate, which creates this precipitin ring. Okay, so this is antigens are here. This is one antibody, another one, a different one, a different one, a different one, and the precipitin is forming between this one and the antigen. Radioimmunodiffusion assays are used to quantify antigens in a different way, and what you do is you measure the size of the precipitation zone that's in a gel. Okay, so it sounds very similar to what we saw. Let's go into in a little bit more detail. So you have insoluble antigens. These are put in suspension. Okay, so it's the solid going into liquid suspension, and it forms flocculants, which are then bound by antibodies. This is the basis of the VDRL test for syphilis, and antitrypanomal antibodies bind to cardiolipin and suspicion. This is the basis of the VDRL test for syphilis, in which antitrypanomal antibodies bind to cardiolipin in suspension. So this is a routinely used test, and again, it basically is looking for complexes that form from antigens and antibodies binding to each other and forming a solid that you can actually see in either a gel or a liquid medium. Viral infections are often detected by quantifying the virus-neutralizing antibodies in serum in this way. Virus proliferation causes cell damage and plaque formation. So for this test, you're looking for plaques. And the way that you do that is you get these culture plates and you put a specified concentration of living cells in the culture plate, and you put a specified concentration of viruses, and you do it in a serial dilution. So you have a high amount, a little bit lower, a little bit lower, a little bit lower, just like we saw earlier. And you get to a point where it's kind of like the opposite of doing a plate count for serial dilution of bacteria, right? What you're looking for, instead of growth of bacterial colonies and making a countable plate with bacterial colonies, what you're doing instead is you're looking for a countable plate with the absence of cells. So the plaque is where there aren't any cells growing. Okay, so these things here that you see that are purple, that is or well, not necessarily bacterial, but a cell line. The purple is those are the viable cells that have been dyed, usually with crystal violet dye. And then the white is where the cells are not there. The plaques are the absence of cells. Okay, so figure 2010, the caption here says, in a neutralization assay, antibodies in patient serum neutralize the viruses that are added to the wells, preventing the formation of plaques. In the assay in the picture here, the wells with numerous plaques contain a low concentration of antibodies. The wells with relatively few plaques have a high concentration of antibodies. So again, the ones that have more antibodies, the cells are alive, and that's going to be these guys. The ones that don't have very many antibodies, the cells are dead, and that's going to be these guys, right? And you can quantify the amount of antibodies, the amount of live cells, by finding a level of plaque formation where you can actually count the plaques and use the number of plaques that you found here and the concentration that was added at the start of the experiment to calculate back how much antibody do you have in this serum. Immunoelectrophoresis is what it's called when you have several different types of biochemical methods. So immunoelectrophoresis is not one method. It's a term that's used for many different methods, but they are all involved in separating and characterizing proteins based on electrophoresis and based on the reaction between the proteins and antibodies. The presence of specific antigens in a serum can be determined using Western blot assays. And a Western blot assay is when the proteins are transferred to a nitrocellulose membrane, and then you can identify the proteins using antibodies that have been labeled with different fluorescent molecules. In a complement fixation test, the complement is used to detect antibodies against certain pathogens. So this is figure 2013, summarizing the process of Western blotting. So this is when antibodies are used to identify specific bands on a gel, which are corresponding to certain proteins. The Western blot is often used to test for antibodies that are fighting off HIV. The top strip is the negative control. So that's this here. The top strip is the negative control, and then this strip is the positive control. And then you have two patient samples that you compare to these. Okay, so this is patient serum samples that have antibodies in them, right? And the Western blotting process, I do implore that you read through this, and I also will post videos of all of these techniques in the description below this video, because I can't really do them justice without making this video like 2 hours long. So for the Western blot especially, but for these other ones too, I implore that you read through them, have a look at the supplemental videos that have animations and things, because they can be kind of difficult to follow, but you need to make sure that you know the similarities and differences between them, what they detect, and the basic mechanisms that are used to detect them. Okay. This is figure 2014, the complement fixation test. So the first step is you have a patient serum that contains antibodies to an antigen that's in the solution. You have another patient serum here that does not. Okay, so this one has the antibodies, this one does not. Both of these solutions have the antigen in them. Okay. The next step, you heat up the serum, and heating it is going to destroy all of the complement in that patient's serum. So now all you're left with here is you have the antibodies that you started with, and then here you don't have anything. Okay. And then the third step, you have an equal amount of the complement that is then added. So you add the same amount of complement to both of these solutions, and then you add antigens to both of these solutions. And you can see in the first vial here, patient A, the antibodies are going to bind to the antigens, and then you have complement fixation that occurs. Okay, but then in patient B's serum, because there aren't any antibodies, there's no complement happening at all. In step 4 here, it's showing that if you have sheep red blood cells added to these solutions, for patient A, the red blood cells and the antibodies are not going to be reacting, because the antibodies are binding to the red blood cells and then they settle to the bottom. Okay, but they don't lyse. In patient B, since there are newly added antibodies there that are not bound to anything, those antibodies are free to bind to the red blood cells, and they read them as being foreign bodies, so they will lyse the red blood cells. And what happens is, if you have a positive for the antibody being present to begin with, then the color of that solution is not going to change even after you add blood. If you have a negative, so you started with no antibodies, then at the end it's going to change to red or pink, okay, because those red blood cells will be lysed by the antibodies that you added in this step here. Right, so let's go through that one more time. If you started with antibodies, and then you add, the first thing you add is heat, okay, and then you add complement, and then you add red blood cells. And if the color of the solution doesn't change, then you know that it had antibodies to begin with. So when you start out, you don't know if it does or it doesn't. But if it does, then it won't change. If it did not have antibodies to begin with, you go through the same steps, but you'll see that the solution changes colors at the end. So this is a nice summary table of the different types of assays that we just discussed. So again, tables are good too. So watching the supplemental videos and putting these things into a table can be very helpful for making sure that you understand the similarities and the differences. But these are the 4 antibody antigen assays that we just went through. We have precipitation, flocculation, neutralization, and complement activation. Okay, and then it gives the mechanisms of how it works, and then examples over here. So we talked about the precipitin ring, we talked about immunoelectrophoresis, we talked about plaque reduction and complement fixation, and we also talked about Western blotting. Okay, the agglutination test, these For the agglutination test, you use cards or microtiter plates that allow for multiple simultaneous reactions to happen. And the great thing about agglutination tests is that you need very low volume of reagents for these things. Agglutination of latex beads is used for what is known as the indirect agglutination assay. And these detect the presence of specific antigens or antibodies in patient serum. Some antibacterial, antiviral antibodies can be confirmed using what is called the Combs test, and it uses what is known as the Combs reagent. The Combs reagent cross-links antibodies in red blood cells, and it facilitates hemagglutination. And then neutralization assays, we touched on this already, but these are used to quantify virus-specific antibodies. So we talked about that when we were talking about HIV just a little while ago. This here is a microtiter. This figure here is showing a microtiter plate. So for scale, this is somebody's thumb and finger. So you can run 96 reactions on this one plate using reaction volumes of like 1, 2, 3 microliters. So it is very high throughput, very efficient. Reduces the cost because you need a lot less patient serum, a lot less of the antibodies and all that kinds of stuff to run these tests, and you can run them simultaneously. This is what an indirect agglutination assay looks like. So these are latex beads, okay? And what you have here is it's testing 6 different types of group A strep throat, and it attaches the antibodies to the latex beads, and each of these are mixed with bacteria isolated from a patient. And if you look here in well 4, you have agglutination, and you don't see that in any of the other 6. So again, it's a case of looking for things that are kind of settling out of solution, things that they started out liquid, they're becoming solid, right? They're kind of clumping up. You see all of the little clumps on this bead. That is indirect agglutination. This here is a nice schematic of agglutination assays and its stepwise procedure. So first you have a blood sample that's taken from a patient that has hemolytic anemia and antibodies attached to their red blood cells. You add to that the Combs reagent. The Combs reagent contains anti-human antibodies, and that gets mixed in with the patient's blood sample. The third step, you have agglutination reactions that are visible. And then down here is another reiteration of the Combs test. So you have patient serum that has antibodies. Then you have donor blood that's added. So that's from a different individual. And the patient antibodies bind to the donor's red blood cells because it's red as a foreign body. And then you add Combs reagent, and then agglutination is observed. So this up here is a direct Combs test. This down here is an indirect Combs test. And for each of these, in order to have a positive test result, you will have agglutination. So you will have solid precipitate that's formed, clumps that are formed. Neutralization assays, so this is giving a much more specific, stepwise explanation of neutralization assays. So what you do is, and this is again in a microtiter plate, you have serum containing antibodies. And in this example, it's for influenza virus. You had serial 2-fold dilutions. Red blood cells were added to the wells, and you can see that agglutination is only occurring in the wells where the antibodies were not concentrated enough. In other words, there wasn't enough of the antibodies to neutralize the virus. The highest concentration at which agglutination occurs is the concentration of antibodies in the patient's serum. And in the case of this test, sample A, right? So sample A shows a titer of 128. Sample C shows a titer of 64. So you have agglutination here, which means you have this kind of solid clump that is forming in these wells, but it is not here. It is not here. Well, there's a tiny bit there. It's not here. So these are negative, these are negative, these are negative. All of these are positive. Positive. Cross-matching tests for antibodies against blood types. So we talked about blood types in the last lesson and what can happen when grafts are rejected in different antigens that are involved in the ABO blood group. So you can see for blood typing, they use this agglutinated clumping phenomenon to determine if you have anti-A antigens, anti-B antigens, if you have both, if you don't have either one. So this one here is showing a really rapid method of typing a recipient and a donor's blood before they perform a transfusion. So they set this up just on a card, and you have 3 little wells, and each one of them is coated with a different antibody. One is an anti-A, one is an anti-B, and one is anti-RH. And then if you see agglutination of red blood cells in either any of these, then it indicates which of the types that you have. Enzyme immunoassays, these are EIAs. Again, this is used to quantify antigens using antibody conjugated to an enzyme. So all of these methods is where you're detecting the presence or absence of antigens, antibodies, and then you're also detecting the amount or the concentration of these things. It just so happens that in EIAs and in all of the other ones, the EIAs, the ELISAs, these are using enzymes to perform these tests. So your enzyme, if you remember from microbiology or general biology, enzymes convert a substrate to a product, and they're not consumed or changed in that reaction. So your enzyme immunoassays are using an enzyme to convert a substrate, which is usually a chromogen or a fluorescent, into a product that you can see. So chromogens impart color to molecules, and fluorescents impart color that glows under UV light. Okay. EIAs include immunostaining, direct and indirect ELISAs, immunofiltration, immunochromatographic assays. Immunostaining is a technique that's used for visualizing cells inside of tissues. So this is your immunohistochemistry. Cells inside of structures like intercellular structures. So this is immunocytochemistry. Immunostaining is also used in histology, cell biology, and also in molecular biology. So you have the next ones we were talking about are direct, indirect ELISAs. So the direct ELISA quantifies antigen that's in a solution. You have a primary antibody that captures the antigen. Then you have a secondary antibody that delivers an enzyme. And the product is directly proportional to the amount of captured antigen. So in a direct ELISA, you can measure the amount of end product, and that is directly correlated to how much of the antigen and antibody you started with. In an indirect ELISA, you can detect the antibodies in patient serum by a more indirect method. So what you do first is you attach the antigen to a microtiter plate. Then you have a primary antibody that binds to that antigen. Then you have a second antibody that will then bind to the primary antibody. Okay, so we'll have another look at that in just a second. Immunofiltration and immunochromatographic assays. These are your lateral flow tests, and these can be used to diagnose things very quickly. Most commonly, they're used to determine if someone is pregnant, but you also can detect other types of conditions and detect, or at least screen for diseases. And this is using color-labeled antigen-antibody complexes that can determine if you have certain chemicals in your urine or other bodily fluids. So let's look at direct ELISA. ELISA stands for Enzyme-Linked Immunosorbent Assay. And ELISA is plate-based, so we're talking again about using microtiter plates, which means, again, that you have the ability to run a lot of these simultaneously, and you can use a very small amount of reagents. And what you have here is, it's just 3 steps. 1, 2, 3. You have an antigen binds to the surface. You have antibody that binds to the antigen, and then you have an enzyme that is released from that reaction, and it changes the color of the chromogen that's in the solution. So it's figure 2022. You use an enzyme antibody conjugate to deliver a detectable substrate to the site of an antigen. Substrate is colorless, and then it's converted into a colored end product. Now, this is an indirect ELISA, and as you can see, there are several more steps to this. So it starts the same. You have antigen, so you put the antigen in the wells, then you have to add what is known as a blocking agent, and then you add the sample. So the sample is coming from the patient, and you're trying to see if the antibody that you're looking for is there or it's not there. If the antibody is there, then it will bind to the antigen that's already in the solution. If it's not there, then it does end up getting washed away in the next step. And then in the next step, you have your foreign antibody that's added. So that's your secondary antibody. And the secondary antibody will add to the first antibody if it was there. Okay. If it was not there, then the secondary antibody is not going to be able to bind to the antigen. Okay, because these don't match up with each other. So if this antibody was not there, then the secondary antibody will get washed away as well. And then in the last step, you add another reagent, and if the secondary antibody is there, the enzymatic reaction will occur, and you'll have your color change. So it's very similar to direct ELISA, it's just got extra steps in between. You have your test solution with your antigen. You are trying to see if this primary antibody is there or if it is not there. In the indirect ELISA, if it is there, it will bind to the antigen. Okay. In the direct ELISA, at this point, the enzymatic solution would be added and you would have a color change. Now, if you're looking for an antibody that will not produce this reaction, then that's why you would use an indirect ELISA, because you can then add another antibody that does produce that reaction and will bind to that primary antibody that you're trying to detect. Okay, so you end up with the same end product. It's just that the primary antibody that you're looking for can't give you that observable reaction. So you put another enzyme in there that can bind with that first one and give you the reaction that you need. Your lateral flow immunofiltration and immunochromatographic assays, these are very simple. They occur on cellulose, so basically fancy filter paper. And they are quick and easy ways to screen for different diseases. They can detect for the presence or absence of antigens, hormones. This figure 2026 is showing a lateral flow test that is used very routinely in over-the-counter pregnancy tests. It is a very simple, straightforward test. If you ever are faced with a positive pregnancy test, you are advised to go and get a more involved screening with your doctor, because that's exactly what a lateral flow test is it's a screening. It's a quick, easy screening test. And things can go wrong to give you a false positive. This is a nice table that is, again, summarizing the types of assays, how they work, and different examples. In your immunofluorescence assays, this uses antibodies that are conjugated to fluorescent dyes. So we have now left the realm of enzymes, and we're looking straight into the world of fluorescent dyes. In direct immunofluorescence, a direct fluorescent antibody, also known as DFA, tests for the presence or the absence of an antigen with an antibody that has been tagged with a fluorescent dye. In indirect immunofluorescence, you can test for the presence of antigen-specific antibodies rather than the antigen themselves. So we'll go through that some more in just a second. In flow cytometry, you use fluorescent monoclonal antibodies against cell membrane proteins, and you use that to be able to count specific cell types that are in a mixture of cells in different cell types. Fluorescence activated cell sorters. This is basically the next step after flow cytometry, in which the fluorescent intensity can be measured, and you can use that to separate cells into high fluorescence and low fluorescence. So let's have a look at the direct fluorescent, the DFA test. This is a really quick and relatively easy procedure, and it's routinely used to identify Legionella pneumophila, otherwise known as Legionella, Legionnaire's disease. So what you do is you stain them with green fluorescent proteins, and then you can use that and dark field microscopy to view the bacteria. And this image is showing a smear from the respiratory tract of a pneumonia patient. So that just involves a fluorescent dye directly applied to a sample taken from a patient. Now, indirect immunofluorescence, this involves using the fluorescent dye and using antibodies. Okay, so indirect immunofluorescence assays, the IFAs, are used to detect antigen-specific antibodies. So antibodies that are specific to antigens that are found on certain molecules, certain bacteria, things that you're screening for. And what happens is it allows the antibodies to bind to antigen fixed to a surface. So this so far is looking really similar to an ELISA. Okay, but there's no enzymatic reactions involved here. All it does is, if it is there, it will attach, and it has this fluorescent tag on it, a flag. And so if it is there, it will attach and it will fluoresce under UV. If it is not there, it doesn't have anything to attach to, and therefore it will not fluoresce. And in this case here, you see all these bright green and yellow fluorescent molecules. This is an example of a micrograph of an indirect fluorescent antibody test, and in this case, the patient's antibodies to the measles virus were bound to viral antigens present. And then you have secondary antibodies that bind to the patient's antibodies and carry the fluorescent molecule. So in English, what this means is they use an indirect immunofluorescence assay to see if this patient had measles, and because it's glowing, it is a positive test. Indirect immunofluorescence assays are used to detect anti-nuclear antibodies and for diagnosing some of your autoimmune diseases. So these are just some more examples in figure 2030. In an anti-nuclear antibody test, you have cells that are exposed to serum from a patient that is suspected of making these anti-nuclear antibodies. And then that patient's serum is then exposed to fluorescent tags that are specific for this antibody. And then you run it with a control. So over here is the diseased patient, and then over here is a healthy control. And you can see where you can kind of see shadows of the fluorescent antibodies that were added. But over here, you can clearly see these cells are fluorescing. Flow cytometry is a way of, they call it immunophenotyping. What that means in English is it is putting antibodies into different groups. And what happens is you have a suspension of living cells. You have stained those cells with very specific fluorescently tagged antibodies. So you're kind of starting out doing the same thing as you did here with the immunofluorescence, right? But the difference is that you're using this device called a flow cytometer. And the flow cytometer takes the mixture of the differently labeled fluorescent antibodies that are now attaching to antigens in the patient's serum, and it passes it through these really narrow capillaries. And then they kind of separate out based on which antigen is present, i.e. which antigen the fluorescent antibody is added to. And then that is analyzed using a laser. The laser excites the different fluorogens. The intensity of the fluorescence is measured, and it can tell you not only which antibodies and which antigens are present, but in what concentrations they are present in the sample. So that is going to do it for lesson 4. I want to remind you to make sure that you have a look through those supplemental videos. Use the tables, again, to help you, because a lot of these methods, they are kind of confusing. They're very similar in their methodology, but they also have very subtle and yet really important differences. So make sure you can compare and contrast the similarities and the differences between these different tests. I want to thank you guys for watching. Don't forget to do the reading. I'm always going to tell you to check the description for more videos related to these topics, and also leave your questions for me in the comments below.